Hi, I'm Lily. Welcome to my corner, and today we're going to be talking about energy, specifically cellular respiration, fermentation, and photosynthesis. So if we're all ready, let's break it down. Okay, so we're going to start off with cellular respiration. So the first step in cellular respiration is glycolysis. So there are two parts in glycolysis. So the first part is the energy investment phase. So that, you can see, is right here. So we're going to start out in our energy investment phase with our lovely little glucose molecule right here. And our little glucose molecule is going to come in and in the cytoplasm. And we're going to add 2 molecules of ATP, which is energy, to the glucose molecule, and that's going to make 2 ADP molecules and 2 of the little phosphate molecules. So that's pretty cool, right? And then we go from the investment phase to the energy payoff phase. Ooh, oh, well, you know what? Our ATP is jumping ship. So in the energy payoff phase, we're going to take those ADP and those little phosphate things that we made earlier, like that we made here. So we're going to wait for another glucose molecule to come along. So we have 4 of them, so 4 each, and those 4 are going to make 4 ATP molecules, which is awesome if they're going to stay together. So I'm just going to put that there. And then we're also going to have 2 NAD plus molecules that come from another one of our processes, and then 2 of our little friends here that are electrons, or 4, can't count, and then we also have 4 hydrogen ions, and we're going to take all those are going to come over here, and they're going to make 2 NADH molecules that will be taken off and used later, so we don't have to worry about them, and also produce 2 hydrogen ions again. And so then all the way down here, our glucose has dealt with all of this stuff being thrown at it, and it's going to result in the making of 2 pyruvates and then 2 water molecules. That, my friends, is glycolysis. Okay, so now that glycolysis is over, we get to move into pyruvate oxidation, which is our next step. And so in this time, our little pyruvates, our 2 pyruvates, have made their way from just the cytoplasm into the mitochondria. So from now on, this is all basically occurring in the mitochondria. So our little pyruvate oxidation, oh, wait, disclaimer. First off, on our model, it only shows for 1 pyruvate, but in actuality, there would be 2. So all of the products that have been made and all that stuff, you would just multiply all those by 2, just so you get the numbers right. Awesome. So our little pyruvate guy has made his way into the mitochondria. So he comes in and he decides to make a bit of CO2 when he's oxidized, so when oxygen's added to him. So CO2 is formed. And then we also have our little NAD plus guy, and he comes in and he takes some of the electrons from our little pyruvate, and they form NADH and then a hydrogen ion. And then our little coenzyme A comes in, and so that forms acetyl-CoA, and so that is the thing because the pyruvate kind of lost its name pyruvate whenever it formed CO2. Then it became acetyl. So we go from pyruvate oxidation, now that part's over once you have acetyl-CoA, and then we go into our citric acid cycle, which is also known as the Krebs cycle because Krebs is the one who discovered it. So we're over here, and our little coenzyme A, he ducks out because he decides he's kind of over it. But that's cool. So we go on, and we go around our little thing, around in a circle, and this motion, the circle motion, keeps happening even after like our 1 pyruvate gets through because more pyruvates are coming. So our pyruvate, well, it's not a pyruvate anymore, it's acetyl-CoA. So it comes in and it loses 2 two CO2 things, and also we have 3 little NAD plus guys, and they come in and they get their electrons out, and they form NADH, and also they take 3 hydrogen ions. So there are 3 of those formed from the NAD plus. So those come in, and then we also have our little ADP, which is this one, and our little phosphate, and those come and they grab some stuff and they form ATP. So that's always good because that's our energy that we like. And then we also have our FAD, which is friends with ADP. It's some of the same type of enzymes. And then those grab some stuff and form FADH2. So that's pretty cool. And then also we have the addition of a water molecule, which helps, and that will rearrange some of the bonds in like the little itty bitty details in here. So yeah, overall, that's pretty much the citric acid cycle. It's called the citric acid cycle because one of the first things it makes is citrate. So that's citric acid. So. Okay, so now that we've finished our little phosphate oxidation and citric acid cycle, we are now at kind of the edge of one of the membranes-ish, and we are about to embark on oxidative phosphorylation, which is a big word. But the model is going to look really busy and really, really threatening, but you know what? We're going to get through this. Okay, so here we go. I'm also not sure how this is going to stay, so we're going to work on it. Cool. See? Yep. That was part of our electron transport chain that kind of flew off there, and my lipid bilayer is not working either. But you know what? That's cool. Stuff happens. So I'm just going to kind of, yeah, we're just going to kind of wing it. Okay. Awesome. So, I'm losing a lot of stuff, but you can see basic concept here, maybe, if stuff doesn't keep falling off of it. So I'm gonna try and see what I can do about this. So basically we're gonna start in, we're gonna start in this corner. Oh gosh. Sorry guys. Well, I'll talk about that later. But we have these big things, and those are our 4 little protein pump complex. They carry the electrons, and our arrows that are falling off, the ones that you see falling off, those are like the arrows showing us where the electrons go. So it's cool. We'll just talk about it. But yeah. So it starts with this little NADH, the one that we've been talking about this whole time. He comes and he's like, hey, I'm full of electrons and I need to get rid of these. So he gets rid of his electrons, so he becomes NAD plus again, and this little arrow that falls off was showing us where he goes. So the electrons go from NADH to this little guy, and this little guy, well, sorry, and this little guy is one of our friends, and he's not really a holding tank, but he's kind of like a mobile carrier. So he's there. And then in our second pump right here, I'm sorry, this is really bad, but in our second pump you can kind of see, so we've got our FADH2 and our FAD. So our FADH2, he's like, hey, I want to get rid of some electrons too. So he sends his electrons to our little friend here, so he becomes FAD again. And so then, yeah, so then our little friend here, our little mobile carrier, he sends his electrons up. There's supposed to be an arrow here, so imaginary arrow to this guy, who is cytochrome C, and so that's a little mobile carrier again, just like this one was. And so this one then moves our little guy down through number 4 here. So his energy moves down, and he joins this little fun stuff going on down here. So if you can kind of see, we've got 2 hydrogen ions and half of an O2 ion. So our little friends who are up in site C come down and join this reaction. So they make an H2O, which is pretty awesome because we always need water. So you also see, like, what's the real purpose of these proton pumps? You're probably wondering. So we're not going to worry about this one yet because that one's different. But all of these have little hydrogen ions that they send up through their little proton pumps. So they all gather, and then they all go to this one. And this one's special because this one is our chemiosmosis. I'm sorry, I'm really bad with names. But so our little hydrogen guy comes through this, and this area, this big area, is our ATP synthase. And so there's also, you can see a little arrow. And so our ADP and our little phosphate over here, they go through the ATP synthase and they form ATP, which is awesome. Then our hydrogen ion just comes out at the bottom, just like, whoa, hydrogen ion. And so that's how it powers the ATP synthase. So that's why hydrogen goes up. And the reason that it comes back down is because the whole point is there's a lot of hydrogen on this side and there's not as many on this side. And so the entire thing with nature and division of stuff on either side of the membrane is due to the fact that you want that diffusion to be equal. So because the diffusion is not equal, because there's so many over here, that's why the hydrogen ions just automatically are like, yeah, I can come back over. So yeah, that's oxidative phosphorylation. So we have our, as a recap, electron transport chain over here, chemiosmosis over here, lipid bilayer that is falling apart. Yeah. Awesome. So that, in its most basic form, is what happens in the mitochondria during cellular respiration. So now we know why it really is the powerhouse of the cell. Okay, so now that we're finished with cellular respiration, it's on to fermentation. And you know what that means? We are about to be 2/3 the way through this video. That's really exciting. So here we go. So fermentation. First off, there are a lot of basic similarities with fermentation and respiration. First off, you need both of them in the human body. So I'm going to show you a little bit of a diagram that I drew up on a chalkboard for us to look at. So what you see in this picture, we've got our glucose up here, right there. We've got our glucose, and our glucose is coming into the cell. So this is the cell, and in the cytoplasm, it's doing its whole glycolysis thing, which is what we just talked about a little while ago, and then it makes a pyruvate. So the pyruvate has two options. Either it can go the way with oxygen, which that would mean it's going back to aerobic cellular respiration, or when there's no oxygen present, it goes to fermentation, which is what we're about to talk about, and that's right here. So with our little thing here, we already know what happens with cellular respiration because we just talked about that. And so over here, we're going to start talking about fermentation, which can produce either ethyl alcohol, lactate, or other products. So what we're going to start with is the lactic acid. So lactic acid is the kind of fermentation that's going to occur when you're working out at the gym and you run out of oxygen to use, so you start using lactic acid. This is also why sometimes your muscles can be really sore after working them out. So it's because of lactic acid. And we know that it's the lactic acid that's being formed, because if it was the ethanol that was being formed, probably wouldn't be as good, because then a lot of people at the gym might be drunk. We don't want that. So we're going to talk about lactic acid. So what we've got here on our little model, we've got our glucose molecule. It's going through glycolysis, just like it did with cell fermentation. There's no change there. So it forms our 2 pyruvates. Also, during this whole glycolysis stuff, we've got 2 ADP and phosphate, and they're going to swoop in and grab what they need, and they're going to make 2 ATP. So that's the energy that we need. So that's always good to have. And then while this is happening, also we have two NAD plus molecules, and those are coming and grabbing the electrons out of the glucose molecules, and they are forming 2 NADH molecules, which are and also 2 hydrogen plus molecules. So then our 2 pyruvates go, and they meet up with these 2 NADH molecules and 2 hydrogen plus molecules. So the NADH decides to deposit the electrons back to the pyruvates. And so this forms lactase, 2 lactase molecules. So that's the lactic acid that we're talking about. So that's how you get energy and lactase all out of our little glucose friend without oxygen. So then our second form of fermentation, which I know is what comes to mind when I think about it just from having a history with cooking, is alcohol fermentation. So with our alcohol fermentation, we have our glucose, just like the first time. It's going to go through glycolysis and form our 2 pyruvates, and our 2 ADP and phosphate is going to come down, grab some stuff and make 2 ATP, just like it did last time, and 2 NAD plus going to come around and make 2 NADH and our little hydrogen ion friends. But as you see, there's a bit of a different stuff over here than there was last time. So what's going to happen is our 2 pyruvate friends, please, confusing. Our 2 pyruvate friends are going to lose 2 CO2, or they're going to make 2 CO2 molecules and get rid of them. So these are going to come out, and then they're going to go down, and so after the CO2 molecules are gone, it's going to make 2 acetaldehyde ion atom molecules. Sorry. But it's going to make those. And then those are going to come, and the 2 NADH and hydrogen ions are going to come, and they're going to meet up, and then they're going to switch their electrons, and so the buddies here get the electrons, and then the NADH form back to NAD+, and our little buddies here become ethanol, or ethyl alcohol, which is always fun. So that is basically our fermentation. So guess what, guys? We're 2/3 of the way done. Now it's on to the awesome, wonderful world of photosynthesis. Okay guys, we're in the home stretch. So now we're going to transition from talking about stuff that happens in mainly animals to stuff that happens mainly in plants. And so I guess you can probably guess what that is. Photosynthesis. So photosynthesis has 2 main parts. The first is light reactions, and the second is the Calvin cycle. So we're going to start with light reactions. So I have a model for this, and you'll see the picture somewhere eventually. But the reason I'm not going to be holding it up is because it's been falling apart like crazy. So I don't want to repeat the last cycle of cellular respiration with all the energy falling off. So we're just gonna go on without it, and I'm just gonna talk about it, and hopefully the picture will appear. So we're gonna start with the light reactions. So what happens is, as an overview, there's gonna be a water molecule. It's gonna enter. Stuff's gonna happen. It's gonna come out as oxygen with the help of light and some input of NADP plus from the Calvin cycle, and the input of ADP from the Calvin cycle, and the output of ATP and NADPH from light reactions. So main parts that you need to know: there are 2 photosystems in light reactions. Photosystem 1, which is the second one that they go to, which kind of wonky, and then photosystem 2, which is actually where we're going to start the action for us. So what happens is light penetrates into the chloroplast and activates the pigment molecules and gets them all excited. So those pigment molecules go up in the little photosystem, and they enter this thing, and they kind of merge together. 2 of them do, and it forms P680. So this P680 molecule is moves up. Technically, there are 2 ways I could phrase this. So I'm gonna do, sorry, but so there's also, we're talking about H2O molecule, right? So it comes in, and it it loses some stuff, and so you see like H2O, and it comes down, and then out here there's a little branch, and it has half of an O2 molecule and then 2 hydrogen plus molecules, and then it goes down, and so it adds 2 electrons to the P680, and then the P680 moves up to the primary acceptor, and it has a little electron in its arrow, so it gains an electron there, and then it goes into the electron transport chain. So it goes through 3 areas in the electron transport chain. There's the first one, which is like this little proton holder, and then there's the cytochrome complex, and then there's another little holder. And so the cytochrome complex, and mainly it's just the cytochrome complex, but the stuff in there forms a molecule of ATP. So that's where we get our energy. And then it goes back right into the primary acceptor, and then of the photosystem 1, but technically 2. And then the light will activate another pigment molecule, and so it gets all excited and joins the other molecule. So they're together, and that forms P70, so P680, P70. So that then moves up to the primary acceptor, and then it's transported to another electron transport chain. And this electron transport chain goes through another little molecule, which is it's a proton thing, and it's called a ferredoxin, so FD. And so then this electron chain doesn't produce ATP, just so you know. So there's, the first one does, the second one does not. The photosystem 2, which is the first one, also has the byproduct of O2 and 2 hydrogens, but photosystem 1, which is the second one that you go to, does not. Just trying to clarify that, but it's probably confusing. So then it goes through there. It'll have 2 electrons that jet out, and then it enters the NAD plus reductase. So that's NAD plus and a hydrogen atom. And so that forms with the leftover type P70 stuff after it's been through all that, and that forms NADPH. And then that's the end of photosystem 1. So just as an overview, what the light reaction produced was 2 hydrogen ions and half an oxygen, some ATP. Well, the oxygen is just for like outside of the chloroplast. Inside the chloroplast, it produced ATP and it produced NADPH. So that's your basic overview. So yeah, awesome possum. Now we're going to move on to the Calvin cycle, which I can actually hold up that model, fingers crossed. So with the Calvin cycle, what we've got here is a pretty familiar looking diagram. You can recognize the circle form from something we saw in cellular respiration, and that's the citric acid cycle. So they're not exactly the same, but there are some differences. There are some differences, but there are also some resemblances in the circle idea. But so it comes in. We've got our little CO2 friend up here, carbon dioxide, and he's going to come down and enter our little Calvin cycle. So in phase 1, he enters and meets up with his little friend Rubisco here, and they're going to be like a team, and they're just going to, you know, join up and be happy and all. And so then we move into phase 2, which phase 2, if you cannot notice, is just, it's very productive. So we have six ATP that come and act on the Calvin cycle and form 6 ADP. So 6 ATP to 6 ADP. And then we also have down here 6 NADPH, which are those full ones. They come in, and that, oh, we lost one. Well, they form 6 NADP. This is the 6th one that fell off. And then they also form 6-phosphate. So that happens. And then also the output here is the sugar. So we see our little glucose friend from earlier. So this way, this is how you can know that plants can also go through cellular respiration, because this is their little glucose. So that's how they can use cellular respiration or fermentation. So that's pretty awesome, right? Because that's like connecting all the dots. I thought it was pretty cool. And so then we also have phase 3 over here, which is still stuff inside the cell. So in phase 3, we have our little 3 ATP that comes and acts in there, and then is put out as 3 ADP. And then we keep going around, and that's pretty fun. But yeah, so as you see, this mainly produced the NADP plus and our friend ADP up here and over there. But it took the ATP and the NADPH that was formed in the light reaction that we just talked about. It took all that and just mushed it in and then produced this, so that we can just keep using it in the light reaction. It just keeps circulating. So yeah, that's what happens. And then our output was glucose, input carbon dioxide. So yeah, there we go. That's our fun little photosynthesis. So now that we've pretty much wrapped everything up, we come to the end of our video and back to just me in a corner. So hope you might have learned something, and I know that this helped me a lot with my understanding of the topic. Hopefully. Hope you enjoyed today's show, and maybe there'll be another one soon. Also, awesome.